دو صفة هي رنجي توريكا لغا شوي. Okay? The shape of the seed and the color of the seed. These are two different traits. Alright. For each trait, we have two phenotypes, one dominant, one recessive. The yellow color is dominant over the green color. The smoothness is dominant over wrinkled shaped. Basha. So we are talking about two different genes, and those two different genes we know from Mendel's experiments, they were on two different chromosomes. That is why they are independently assorted, and that is why you end up with a classical 16-part Punnett square. Okay, so we start there with parental generation. Okay, through breeding, so you have two homozygous. They are both homozygous for those two phenotypes, for those two traits. So you have a plant with yellow, smooth seeds, and another plant with green, wrinkled seeds. Okay, you do the crossing دائما بزمن لا ستاتيسيشن ثاني يعني هالشيء اللي امتحانات inheritance crossing or tanker, you must always do the gametes. دائما gametes كان جاكنوا. كيشوي crossing كان ليش هذا مكان؟ باشا؟ Okay. So what sort of gametes can this individual this plant has for those two genes? It could only be Y capital, small capital. Y capital, S capital. Alright? Do you understand this so far? Right. And for the other one, it could be only Y small, S small. These are the gametes. Those two individual plants might only have those gametes. Then, of course, as a result, the result is going to be plants with yellow. He had plants with yellow, smooth seeds. Now, when we do this, is the F1 generation, filial 1 generation. When you have this heterozygotic plants, you cross them together, then you separate the gametes. What possibilities of gametes do you have? You can have Y capital with S capital, yeah? You can have Y capital, you also can have S small with Y small. Pasha? But you can also have Y capital with S small and Y small with S capital. This is what it means by independent assortments, that the alleles of each gene are independently assorted if they are on different chromosomes. That one allele of a certain gene could end up with one allele of another gene or the other allele of that other gene. Asha? Okay, so here we've taken two examples, it's easier. So when you do the crossing, and this is Mendelian experiment, when he did the crossing, you do the crossing between the two, you have all those gametes. The gametes could be Y capital and S capital and all the other possibilities. With the other plant that you're crossing with, it is the same. Haman أو جوري gametani هي أو plant you cross here. The F1 generation. شكتوخذ two plant crossing أو من the two plant crossing the F1 generation. كأزاني already heterozygotes عشان كلا P generation بولس كده. باش. أم جوري gametani ما هي لها plant كان. crossing possibility كانت ولا كان probability كان. So Y capital S capital with Y capital S capital. This is the pattern. And so on, you do all the crossing. And you end up with the typical 9/16, which is all, all capital, which means all dominants, and 3/16 having been heterozygous, some dominant, some recessive, and 1/16, which is all recessive phenotypes. Okay? So this is an example of the independent assortment. When it comes to diseases or syndromes, some can be inherited as dominant, autosomal dominant, some autosomal recessive. Here are some examples in this table. Be familiar with some of them because you will come across them anyway. So, for example, you have hypercholesterolemia, I've written it in the text as well. You have spherocytosis, you have Huntington disease, and so on. All of these are inherited in dominant form. This is an example of a crossing of dominant. We're talking about diseases here. So if you have an affected father, which is heterozygous, affected but heterozygous, so he has one recessive, one dominant allele for that particular trait, and the mother is unaffected, you end up with this pattern of children. Because this is an autosomal related trait. It doesn't matter. You could, if you have the information, you can write it down, but it doesn't matter, okay? Of course, the pattern will change if you have a father. For example, if you have an affected father who is homozygous, what will be the result? Yeah. All the children will be affected because it's a dominant trait, a dominant phenotype, even if the mother is unaffected, and so on. Of course, it changes based on how you start. Some diseases are inherited in recessive form. For example, cystic fibrosis, phenylketonuria, alkaptonuria, and some sickle cell anemia and thalassemia. Those to be familiar with particularly. Again, here is an example. If you start with two carrier parents, وشي carrier لحالتنا خوشي لحالتها بكارينية بو كهينة كخوي recessive. If you start with two carrier parents, these are the possibilities of their children. Of course, these possibilities changes if you have the parents being different, both of them affected, none of them affected, one carrier, one affected, and so on. But we have other patterns of inheritance, and the reason is it happened by chance that Mendel studied traits which were controlled by alleles, by genes that have two alleles, only one gene and two alleles, and each gene responsible for a trait was on different chromosome. But we know now later on it was found that genetics or inheritance is much more complicated than that. These are the major other ways, other forms of inheritance. Okay? So we have بدلبن أو جماعتها أما تشجع حاضرة أو جماعتها تشجع بيتها أو بدلبن حتى أجر مناقشي بابتي أيوة حتى أجر مناقشي بابتي محاضرة كأن استنى تركيزي خطة الخطي في شيء كذا. Okay? This is the very last time I tell you. Next time you have to read the lecture. I'm sorry, I don't mean to be strict. All I'm trying to do is make you concentrate with me. That's all. Okay? I know it's a little difficult when you're sitting and listening to someone to always concentrate, but you must try and help me. Alright. But then, so we have X-linked. Why is it different? I mean, it is the same pattern, the X or Y, like autosomal chromosomes. It's the same thing. However, there is one difference. And the difference is it is related to gender. So being male or female matters there. Okay. Y-linked inheritance is very rare because the number of genes on the Y chromosomes are very few anyway. We have codominance, incomplete dominance, multiple alleles, linkage, epistatic, pleiotropy, polygenic, and continuous.  'cause continuous variation, and we also have other factors that affect, the inheritance. Okay, affect, environmental factors that affects inheritance. Okay. So the majority of genes, when it comes to the sex linked, are on the X chromosome. The X chromosome is a large chromosome, and it has a large number of genes on it. Okay? And Y is very few. In this diagram, there is a mistake, but it's okay because the Y here it should have been drawn much smaller because it's a very small chromosome, the Y I've shown you in the image before. Anyway, it just gives you an example. If there are one gene with 2 alleles on the X chromosomes, of course, when you draw those genes in a male, it is going to be on the X chromosome, not on the Y chromosome. هرچی جاره و گفتم پیتن هرچی جاره تو کیسه که باید کیسه که this gene or this trait is controlled by a gene on the X chromosome. دائما نبي نسال X كو ولا ينرمز جينا كبابين R شيا أي هرتي كذا نسال X كذا يعني أو راسا نبي نسال Y كذا يعني مش كمية نسال واحد ولو بيشوانا ولا نسال Y كذا نسال X كذا يعني. The word linked اللي ربى كاري هنري ما بس أولية linkage كlinked كيسدي الباسية أو يكدو جينا سنة كروموسوم أو اللي بس هل جينك لسه X chromosome كده أو purely gender linked or X linked or sex linked, okay? And this is what I've written here, right? I'm going to explain it. So we have X linked dominant, we have X linked recessive. X linked dominant is, here are some examples. For example, here we have parents. If the father is affected, because the father is going to be affected even if it's recessive, because males have one copy of the X chromosome. Okay? So if you start like this, the mother unaffected, the father is affected, here is the pattern. If you do the crossing, there is a pattern of the children. Okay? You have the sons unaffected, the daughters affected. Why? Because the daughters are 2 X's. They get one of the X's from the mother, but they must get the other X from the father. And since in this case we're talking about dominant case, the allele that causes a certain disease is dominant, it's going to appear on the daughters. And here, this is another example. There are many examples. One example is the fragile X syndrome, where it's actually a part of the X chromosome itself is like fragile, and many genes on it are affected. So it affects few genes on it. Okay, and X-linked recessive, the famous example is color blindness. Again, you can see the father is affected even though it's recessive, because males have one copy and it's going to appear anyway. And here it shows you 2 examples of, if you have a mother unaffected or carrier, how is going to affect the pattern of the children. Color blind people, they don't see subcolors. There are degrees of color blindness. It's actually the gene responsible for the protein opsin, which exists on the retina, the membrane of the retina of the eye. If it is Y-linked, it's very easy. The pattern is very easy, inheritance patterns, because all the males are going to be always affected, and it's not going to affect the females, of course. Codominance is when you have both alleles having their phenotypes shown together, not mixed together, but together, as you can see in that flower up there. Both phenotypes of that trait affect, that is codominance. Okay, and a good example in human is the ABO blood group, where A and B, both of them are dominant, whereas O, responsible for by the gene I, small I, is recessive. Okay, and that is why you have people with blood group AB, right? Both of them are dominant. Both of them are, they show their effect. multiple alleles because we have 3 alleles for this gene. A, B, and the one responsible for O. Incomplete dominance is when the 2 alleles show the mixture of their effects. So the phenotype is a mixture, not both of them show together. As you can see here, one allele is responsible for the color red, the other is for the color white. The mixture of 2 is pink. زين لو كنتي هردو رنجة كذلك لذا تيكلا يهردو رنجة. This is incomplete dominance. An example in human, there are many examples, but wavy hair is considered a good example between straight hair and curly hair. Wavy is a good hair. I want to get your attention into this. Look into this square here and see how the inheritance in the Punnett square is written. When do animal heredity dominant negative recessive shaking, a chin, Peter capitalaka, Bacardi bo dominant, to smallaka bo recessive. Alright? إذا شغلت بحالة shaking. Not only our capital is small. Lena لحالة أبي دو بيت Bacardi. باشا؟ طبعا دو بيتكا شو Bacardi؟ يا وكو أحالتها Tolkite color shade away another ball gene okay, ball color okay. See the kind now shade color. Like it can be shade color. When the cell or ya R ball red, ya W ball white, have you keep a capital, have you keep dominant? Bacardi now. Another halati co-dominance is shwasta Ya atwani okay, ya agarhatas yakana each organa inherit cell. Babita capital canvas okay. So in the case of codominance and incomplete dominance, you must use 2 different letters of the alphabet. Otherwise, it will not be right. Once you do that, you can do the crossing. Another example of incomplete dominance is Tay-Sachs disease, where people lack an enzyme that breaks down a lipid, and it leads to the accumulation of this lipid and nerve cells particularly, and it leads to physical and mental disabilities. But although, we can say there is 2 patterns of inheritance. Although it is inherited as autosomal recessive, however, in a certain percentage of the population that is being studied, it's been found that those people that have 1 allele, they produce only half of the amount of that enzyme, that protein. It's supposed to be, because it's recessive, so individuals, when they have 1 allele, 1 normal allele, non-mutated allele, they're supposed to produce that protein and have no problem. However, it's been found that in a large population, over large study, they actually produce half. So in this case, this disease is showing also a case of incomplete recessiveness, but also incomplete recessiveness is incomplete dominance. Incomplete dominance, okay? So this is an example of incomplete dominance. Okay, linkage. Okay. When 2 genes are on the same chromosome, 2 or more than 1 gene, 2 genes on the same chromosomes, they break the law. They don't go with the laws of segregation and independent assortment. They are linked genes and they co-segregate. So here, if you have 2 chromosomes and these 2 different genes represented by different colors, when they are on the two different chromosomes, they are going to have independent assortment and segregation, as I have explained. But if it's on the same chromosome, they are not going to have independent assortment. The alleles are going to be certain alleles are always together. There are many examples, but one example which I think I'm going to give that question in the study session is hemophilia and colorblind. So color vision is a trait. The color blindness is the other allele of color vision. It depends on what type of obscene, the presence of all three major types of obscene proteins. Anyway, hemophilia is another disease, it's something else. But because the genes responsible for those two traits are on the X chromosomes, they are on the same chromosome. That's what's important. Because they are on the same chromosome, those two genes are linked. And yes, this is how we see the pattern. Usually people who have hemophilia, they have color blindness as well, and so on. On the upper part here of the diagram, it shows unlinked genes. Again, we're seeing this again. If you have two different genes on two different chromosomes, represented by different colors, they are going to be assorted in all possible ways. It could be A capital with B capital, A capital with B small, B capital with A small, and so on, or A small with B small. All the possibilities, independent assortments, and you end up with the 16th part of the Punnett square. But if you have linked genes, those two genes, A and B, are on the same chromosome, not on different chromosomes. There's going to be only two possibilities of the gametes formation of the alleles. For example, if originally A capital is with B capital on one of the homologous chromosomes, the other homologous chromosome, let's say 2 chromosome 1, is going to have the B small and A small. So the recessive alleles are going to be always together and the dominant always together. Okay? And this, you end up with smaller probabilities in a Punnett square. And it's going to be 3 to 1. The person who, many people studied, Bateson and Punnett studied and found out about linkage. However, the one who studied it extensively is Thomas Morgan and his PhD student, and he's a Nobel Prize winner. They studied these things on Drosophila, fruit fly, and Drosophila is one of the organisms that is extensively still used in genetic experiments. So he found out some of these examples, some of these traits, they're always together. So the fruit flies, the Drosophila, which have the gray body, they always have, with the gray body, they have red eye and large wings, for example. And those with the black body, they always have also small wings and brown eyes. Okay? So these traits always come together. So we concluded they must be encoded by genes on the same chromosome. But to make your life more complicated, we have something called crossing over. Again, crossing over was found by Catholic priest Franz Jensen. He discovered crossing over, but again, this was confirmed and discovered well by Morgan. So crossing over and recombination, the same thing. They can end up in making two linked genes unlinked, depending on the distance between them. So even if there are two genes on the same chromosome and they are linked, their alleles are going to be always, linked, as I explained. But they can separate, the alleles can separate, so they can have different combination by the process of crossing over that occurs in meiosis 1. A reminder here, a small reminder is, this is the exchange of pieces between homologous chromosomes in meiosis. Let's go to this image again and try to go into a little details. These are all chromosome This is paternal chromosome number 1. This is maternal chromosome number 1. Okay? Look at the X, Y, and Z. Imagine these are they represent 3 different genes. هذه بيربرسن الشركة. بو آسان كاني خوي رنجي تشاو رنجي بيست أو آسانية بلام بو آسان كاني بوي خيالي كان. بابلين إكسكا رنجي تشاو وايكا رنجي خجا زيدكا رنجي بيستا. باشا بو آسان كاني ما جينا كان باش. ليرة كابيتالا كان لباترنا. لكروموسوم يكيتري small كان. So the recessive alleles of each of these genes, responsible for those traits, exist on the maternal and the dominant on the paternal. Could be the other way, doesn't it? Okay. Once you end up, now before we go into crossing over, what's the crossing over then? Agabili era. أما دوايوي يعني ما يوصل طوابع هل جامعتك من يكي لمعنيتها يا أمان يا أو يا سورة يا بتقالك يا زرك طبعا أمسور بتقالك زرد صوزتشيا chromosomy paternal chromosome number 1 is composed of 2 sister chromatids because this is after replication before separation chromatid هل أولى DNA كدبل هو replication هو okay لقمة I hope so. And this is 2 sister chromatids. When we do the gametes, الجامعة كان جاي توجيه به. هل الجامعة كان أو individual؟ هل الجامعة كان أجل هي أميتها به، أجل هي أوبي. طبعا أمبي أوبي وقتها مو dominant form مكاني هي. أجل هي أمبي يا أوبي وقتها أجل هي recessive form مكاني هي. ماشي؟ So there are 2 possibilities for the gametes of that individual. Either he has the gametes which contain the 3 dominant forms of those genes, or the gamete that has the 3 recessive forms of those genes. Right. And they are linked because they are on the same chromosome. So that means if you have another such individual crossing over. If you have this individual marries another individual, the same gametes, it has to be the same gametes, human gametes, okay? What happens? There are only 2 possibilities for the phenotypes. Either the children will have dark hair, dark eye, and dark skin, or all of them fair, yani color. Right? But crossing over makes things complicated. The points where the crossing over is a chiasma, what happens is that alleles are exchanged in crossing over because crossing over happens only between homologous chromosomes. So look at the gametes here. Say gametica chiana. The gametica chimanhea. X capital man here, balang Y small man here, Z small man here. The gametic itra Y capital man here, legal X capital, legal small z, and so on. Crossing over makes other possibilities of linkage possible. Okay? وتألي لدومينانت كان بيكا ونما نواليتر هم بيكا ودوسيس فكان بيكا ومنوا. واي كل ألي لكان جوازية تيجا مبسجيا crossing over جينا كان نادوري جيني أي بيانات شيني جيني بي لا أو شترا أو كروموسوم الأبنورماليتي أو عينا قمانية إنه باسي بروسيكي أسايي اعتيادي أكين كهودا باشا crossing over ألي لكان غوريتوا لبيني homologous chromosomes باشا واي ميديا أم individual أم gamete here لقد individuality تلك أم gamete عليها أجل هي من الله كان يان combination زوريان به رجعتش يان توبه بلام تشاليان سكالبه بيسيان كالبه يان بيسيان توبه رجعتش يان كالبه تشاليان كالبه and so on. I think this is a bit more clear now. Okay, so this is what crossing over answer. Alright. Now, if we look at here, what we need to understand is that measuring of recombination frequency is done by knowing the number of recombination in a phenotype in a family history, as you will see in the study session, and the total number of progeny since the appearance of that recombination. It's not complicated to measure it. Okay? The lower the recombination frequency, the more closely those genes are physically. Because we need to remember something. Crossing over can separate the alleles of genes only if they are far from each other. But if they are very close, like in the example of genes B and C, crossing over cannot separate them. That means capital B will always remain with capital C, even again crossing over with So capital always with capital of those two capital dominant forms of those two genes. But A with B or A with C, yes, the alleles can have all this difference. باش تسيرك تعميركم شتيتوا أنا ما أخوي بس أمريكا زي تكتيزي أو كانت الأسان يزور طوالي شن دقيقة ليش وانيا تسيرك تعميركم باش إما علي أو يسروا أنمي جينز أو يكا أو possibility أنا A capital B capital أو هم اللي كمي قامت كنا then we do the crossing we end up with this Punnett square. أنا إخوار ويش أقدر جنبي أو هايكي كوش هرم possibility من هنا باش إذا حالتك كان لين جنبي بلا crossing over ودا possibility كان شون باي باش شيء أنا تو أنا بالضبط هو سوابل باي أيضا شيء كان جواز بأخو possibility can. Okay? K3. Anyone else, please? بلي أخو. باش. إما حسابك دورة. أجر دور بالليل مسافة زورة بين بين. كروسية كيان بوكي. وكو أوي سروا عليه بلي. باش إيه generation يدوهم إيه إقبال أي شيء كم possibility هي شوالة يعني ممكن أما أمانة أخذنا شوالة بلي أبيت الشانسة okay you have all come together to the right answer. Possibility of chance again. What do we massacre? But Bala وقوتت لراوي تفيشو تدخش possibility كان يجوازه بو يعني جمالي possibility كان probability هي possibility كان جوازه ليش chance أبيه أوكي بلان probability هي كأول كان جوازه بو چونك إيه مشيك معود crossing over excuse me and combination is a rare process وتابهمان راجي قامت كان يتقدر ناكري اوكي انجا شيك ما نبيه نشي باقي على السلامة اوكي اليرا إخو إما ألين crossing over باقلين crossing over بو أمسيجينا وريدا إخو معناه وين نيا بو أمسيجينا لهريك لقامت سيلا كان أمريدا تو كقامت سيل دروسلي إخو دوو گامێتسەڵ دروست نابێت چەندەها germ cells they go through meiosis to form a large number of gametes. واتە ئەگەر هەتاکو بۆ ئەم سێ جینە ئەوکەوەرگین لە هەمی لە گامێتەکان crossing over ڕوویدا و عادەتەن لە کەمینەی گامێتەکان بەڵام لە گامێتەکانی تری کە زۆرینەن crossing over ڕووی نەداوە ئەمانە وەکو حاڵەتی یەکەم کە بۆتان باس کرد ئەوە بمێننەوە کاپیتاڵەکان هەموو پێکەوە بۆ ئەم سێ جینە سمڵەکان هەموو پێکەوە دەکەن باشە لەبەرەوە کە تۆ کرۆسینگ دەکەی لە پەنسفێرۆانە تۆ ئەبێ ئەو حیسابەش بکەی کە ئەو ئۆریجینەل پۆسیبڵیتیەکە کە زۆرترە ماوەتەوە واتە وەڵامەکە ئەوەیە بڵێ وەکو سیکستینەکە ئەگەڕێتەوە when it is linked but there is crossing over it goes back to the 16 parts but the probabilities differ not وەکو سیکستینەکە نییە بڵێن in 9
باشە خۆی ئەسڵەن ئەمە مهمە شتێکی وانەمە شتێکی وانەما چکەوەی تەمامە زۆر ئاسانە پۆلیجەنیکە وەنیها سەمەکەن با تەواویکەین حەزەکەن چاوڕێکن کارەبا بێتەوە بێدە باشە بێدەنگ بن ئەتان بینن قسەیشتان بین ئۆکەی لە هەمووی خۆشتری نەکەم کردووە بێدەنگ بن polygenic when you have more than one gene responsible for a trait, like in the case of eye color, for example. Pleiotropy when you have one gene, but this gene is responsible of the appearance of many traits, like Marfan syndrome, because it's responsible for one protein that exists in the connective tissues. Connective tissues exist in many parts of the body. You also have multiple alleles basimtir, you have environmental effect. Environmental effect is known because nutrition, lack of sleep, lack of exercise affects the height. And also exposure to the sunlight affects the color of the skin. Chitri Mayawa, the Jorakani inheritance. Epistatic, Bashtirin Numune epistatic is when the effect of one gene covers the effect of other genes. Born Okiawaniwa Kachalan, Iko Ojinean hair, Karangi Taibeti Habibo Kushian. Balam Aslan Laberawi Kushian here. Laberawi gene يكتر percent boldness لكتشالي وأكا أوكي جينكي تري رنكي درناتج فأصلا كجكيا so the effect of one gene covers the effect of other genes. I think that's all.